So,
Can you hear now, all of you? Okay, great. Then anyway, what I will do is so hopefully everyone is safe, fine. Everyone is doing okay at your own places, and this is our first online class. I think we'll be spending little less this time. Maybe from the next week class onwards, we can do little bit longer. What I'm trying to do is most of the classes I'm recording. If the records are fine, I will try to share it in YouTube or the site itself. I will make it out and then share with the people. Okay, so today what we are going to talk is mostly the things I have shared with you. We'll go ahead with it and then we go forward in the next class onwards. So what I have been sharing with you all is regarding some of the non-coding RNAs. And non-coding RNAs as such have become very, very important. Maybe equally important like the protein coding genes. So in the case of non-coding RNA, as you can see in this slide, what we see is that it can be classified into two RNA. One is the structural non-coding RNA and regulatory non-coding RNA. As you can see here, non-structural or structural non-coding RNAs, as we all know, is classified into rRNAs and tRNAs. We talked a little bit about it during our protein translation classes. And mostly these two classes were well characterized and has been studied well, and the structures, even in the ribosomes, have been solved. So what we are going to do in the next few classes is probably including the next class, what we are going to do is we study about the basics and little bit the role of regulatory non-coding RNAs. And in this regulatory non-coding RNAs, I have been sharing you a few of the papers, including long non-coding RNA. And I have shared papers regarding microRNA. And we have something, so one of the papers review as circular long non-coding RNAs has been shared with you. So today, we'll just start with something with long non-coding RNAs. Before we go ahead, the regulatory non-coding RNAs is classified or divided into three classes. Depending on the size, it has been classified into three classes. One is the small non-coding RNA, which ranges from 20 to 50 nucleotides. When you say around 20, basically it falls most of the microRNAs, then PV interacting RNAs, siRNA, and then telemeric sequences. All these things falls into this category. And the next class is the medium range, so medium classes, which spans from 50 to 200 nucleotides. And this comes with small nuclear RNA we talked about, then small nucleolar RNA, RNAs belongs to this class that is the medium classes of non-coding RNAs. And in the case of long non-coding RNAs, the interest has been, some of them are discovered, some of them were discovered quite some time ago, like the X inactivating schist RNA that we discussed in our non-coding RNA, which helps in the X chromosome inactivation. So those ones, for any of the RNAs which are more than 200 nucleotides long are classified into long non-coding RNAs. But what is known is that in the long non-coding RNAs, again, you can have different varieties of, depending on the biogenesis, depending on the structure, and depending on the action, it can be classified into different types of RNAs. Okay, going ahead with the long non-coding RNAs, what we are seeing is that depending on the kind of product or the genomic regions you are getting at, it can be all different types of long non-coding RNA. For example, in the case of first one, many of the long non-coding RNAs are transcribed from the enhancer region or promoter regions. So in the case of enhancer regions, you can find just it can be in both directions. There can be both directions of the enhancer. You can have different types of long non-coding RNA. And one very important is the divergent linked RNA. Divergent, why it is called divergent is that it produces at the same locus, it produces two RNAs which are using the same promoter region. It is transcribed in opposite directions. Two of them are two of the RNAs are transcribed from the intergenic region. Okay, and then when we talk about overlapping, what does long non-coding overlapping RNAs, what does it mean is that the long non-coding RNA which is being transcribed spans from coding region, that is the exonic region, these black bars here. The exonic region and intronic regions are both transcribed. In one way, overlapping long non-coding RNAs means that it contains both intronic sequences as well as the exonic sequences in the same long non-coding RNA. So as it can be said, it is a mix, not truly intronic, but when it is transcribed only from the intronic region, we have intronic RNAs. Okay, and then depending on the direction it has been transcribed, because many of the long non-coding RNAs are transcribed overlapping the protein coding genes, that is PC genes. If it is in the same direction, it is the sense RNA, and if it is in the opposite direction, then it is the antisense, as you all know, antisense RNA. Okay, so you have the both sense and antisense RNA, and if it is in between two of the protein coding genes in between, that is the intergenic region, you will find many of the long non-coding RNAs in this. So I want to add that all these, if you look at it, it can fall in the protein coding regions, or it can be in the enhancer region, it can be in different directions, and it can be in between two protein coding genes. So if so far what has been reported is that if you do genome-wide association studies, genome-wide association studies is something like the particular SNP, that is single nucleotide polymorphism, is checked whether it is linked or it is represented more frequently in a disease condition or as compared to normal healthy conditions. So in many of the GWAS studies, what it has been seen is that most of the linked SNPs are found in the non-coding regions. So there may be good reason to think, I think the way forward to look at is that many of these non-coding RNAs might be falling it here, and it may, if we look into the long non-coding RNA or non-coding RNAs as such, may be very good way to look forward to understand the phenotype of those linked SNPs for disease conditions. So I think that is way forward we have to look at. And then furthermore, what if you see it is again summarized or in pictorially it is shown here again what kind of RNAs, long non-coding RNAs is there. It can be at the enhancer region, we call as eRNAs, and it can be in the promoter region, promoter associated long non-coding RNAs are there. And exonic regions have been seen, which can be both in the sense or anti-sense direction. And in the intronic region, you can see sometimes you can see intronic long non-coding RNAs. You can find many in many conditions. And as we talk about in the earlier, when you have a divergent body, what you can see is that there is a short overlapping sequences here, which can complement each other to form a kind of a complex of two long non-coding RNAs. This can be found in many of the regulatory long non-coding RNAs. Of course, you can see in many times you see in the long intergenate long non-coding RNAs as well. As I talked to you, it can be antisense or it can be sense long non-coding RNA. Okay, so depending on this, there are different types of long non-coding RNA. What it can be seen is that as we discussed just now, there are many long non-coding RNAs which are in the intronic region, that's called intergenic region, that is long or large intergenic non-coding RNA, linked RNA. These are transcribed from the intergenic regions between two protein coding regions, and mostly it is transcribed by polymerase II, which transcribe the mRNA as well. So most of the time you get the 5' capping, that is the M7G capping, as well as polyadenylation. So what it has to be noted here is that most of the long non-coding RNAs are derived or transcribed using RNA polymerase II, and as such, like any other mRNA, most of them are M7G capped at the 3' position, and it is polyadenylated. You can see here. So long intergenic non-coding RNA linked, and then you have the natural antisense transcript or NAT, which can also transcribe by polymerase II. And this is one of the examples wherein after transcription, what has been seen is that long non-coding RNAs, many of them are processed post-transcriptionally to make it stabilize or to mature itself. Just like we have the tRNA maturation in the case of non-regulatory or structural RNAs, tRNAs, the RNase P recognize certain sequence in the transcript as well as the structure which is followed and then cleave at certain position, which can give rise to long non-coding RNA. For example, in the case of MALAT1, this is processed by RNase P, and there is a triplet structure, UAU structure is formed, and that doesn't allow the degradation of mRNA, because this RNA, MALAT1, doesn't have poly A tail to protect it from RNAs which would degrade the RNA from the 3' position. MALAT1 forms a structure, triplet structure, UAU structure, which stabilizes the long non-coding RNA. Then followed by, if you can see here, SN or RNA, many of them are derived after transcription. It is processed again, just like the RNSP processing. You get the SNO link structures are there, and these are very similar to SNO RNA, but it is related, and these structures are the signatures of the small nucleolar long non-coding RNA, which is again after processing. These structures resist the long non-coding RNA from degradation. Then you have many times you have other RNAs which are mature from the normal mRNA, and one more stuff which we will be discussing, like the circular RNA, in the next class. What we can see is that after splicing, uh, what you get is the lariat structure. Okay? So the lariat structure as such is circularized RNA. So many times these circular structures are processed further by RNases, so that the 3' overhang in the lariat structure after splicing is processed and results in a circular RNA, which is resulting from intronic regions. Okay, so now the intronic regions can give rise to circular RNA, the stabilized circular RNAs. In other processes, what it can have is that the if you look here, the intronic regions which are green here, what it can do is that generally what you get is one intron donor. The donor site, which is at the 5' side, can ligate or target the 3' donor site. These part of the intron give rise to lariat structure. In many of the transcript, what it can happen is that the it can ligate one of the donor downstream and ligate with the 5' side of another intron. So when you have this splicing, gives back, we call it as a back splicing. So 5' side ahead splice with the downstream, that is the back splicing happens, or one of these donor, the donor splice with this. Okay, so there is a directionality change and it can result to back splicing, and this back splicing results in the proper circular RNA as such. So these circular RNAs have been discovered quite some time ago, and it is known to be playing a good role in gene regulation, that we can discuss how it is functional. Okay, so then going ahead, there are a lot of long non-coding RNAs which have been discovered. This is just an example. It's not the limited. It is a limited example that has been shown to be there in many of the organisms. For example, X inactivating cyst, long non-coding RNA, that we talk of X chromosome inactivation, which are seen in mammals. And you have even in invertebrates, in worms, you can see some of these long non-coding RNA. And long non-coding RNAs, the transcriptional units are seen to be conserved, but however, if you look at the human long non-coding RNAs, around 30 to 20% are very, very unique to humans. And if you look at a sequence conservation, it's no more conserved, or it is very much related. So what I want to say is that the around 70–80% the transcriptional regions are very much conserved in the case of long non-coding RNAs, and around 20 to 30% are, mostly around 30% is very unique to the organism. For example, humans or mouse, it's very much unique to it. And what has been observed is also that the long non-coding RNAs or other non-coding RNAs constitute majority of the transcripts, as we all know. So one is the structural long non-coding RNAs, like the RNA, tRNA, constitute 80% of the transcript. Then if you look at the protein coding region in the whole genome is less than 2% or around 2%. Now it is believed that the non-coding RNAs, including the long non-coding RNAs, constitute majority of the rest of the RNA that we see in the transcriptomes. So how does these long non-coding RNAs are important is just evolving. People started looking at it, and it's of much importance to look into the regulatory aspect in case of cellular differentiation or tissue development or the development stages specification. All these things are looked into. So if people, when people look into the long non-coding RNA's function, can be broadly fall into three types of functions. One is it modifies the uh, chromatin structure either by recruiting uh, the transcriptional activation proteins or RNA proteins, or mostly the change in the epigenetic pattern of the particular locus. So one function is the change in the chromatin structure, either at the histone level, very basic organization of chromatin structure, or DNA organization in the case of eukaryotes, or it can uh, recruit certain factors which binds to RNA, and that affects the epigenetic pattern or the packing of the nucleosome pattern. All these things can change. So one is at the chromatin level, it can regulate the structural change in the chromatin organization. Second thing is it can change the transcriptional level by recruiting activators or by recruiting the inactivators and change the pattern of transcription. For example, change in the transcriptional inhibition by sequestering the inactivator. So one of the examples, what it has been seen in many of the immune systems, is that you can one of the inactivators is sequestered by long non-coding RNA and doesn't allow the inhibition to happen. So this kind of things has been seen. And another thing is the long non-coding RNAs serve as a precursor for many of the smaller non-coding RNAs. For example, microRNAs are transcribed as a longer precursor that is processed, and it can be uh, uh, it can give rise to microRNA after the processing. That's one. And then third thing is the or fourth thing is the mimics the uh, mimics or sequesters the factors. For example, uh, the microRNA targets can be protected when some of the long non-coding RNAs are transcribed in such a way that these long non-coding RNAs have target sites. So it sequesters the factors away, or microRNAs away from the real or regulatory genes that is supposed to target. Then when it comes to, for example, as we talked about how the chromatin factors, chromatin organization is changed. For example, the H3K4 methylation pattern or H3K6 methylation pattern affects the RNA polymerase activity. And if you look at it here, what it has been seen is that many of these H3K K4 trimethylation is recruiting RNAs, non-coding RNAs that would act in the activation of polymerase 2 mediated transcription. So many of the enhancer eRNAs, enhancer binding RNAs or enhancer element RNAs, eRNAs, can give rise to the change in the chromatin structure in such a way that the RNA polymerase 2 can load on the proper site and activates the transcription. And another way to go is that you can get ready the enhancer element, mark the enhancer. When the time comes, certain factor comes change, and then that is recognized by certain factors. And it's not always true. The other form is that you can have repressed enhancers as well. These RNAs can recruit and condense the chromatin in such a way that the transcriptional unit becomes inactivated. So long non-coding RNAs can act in all the ways, that is, it can activate, or it can poise the enhancer element, or it can replace the enhancer element depending on the epigenetic level or histone organization, and by recruiting or by not allowing the transcriptional factors to bind to certain sites of the genome. Okay, so to summarize, this would be the last slide for today. So uh, what it has been seen is that the long non-coding RNAs can act in very different level, at different level of the organization. As we talked about, the uh, at the chromatin organization state, the cis RNA, as we talked about, cis RNA mediates the binding of certain factors and the laminin proteins and allows the condensation of the X chromosome around the nuclear periphery and inactivates the X chromosome. Okay, that is the first level. This affects the particular, it starts with a very basic chromosome level or nucleosome organization, and it condenses. Second thing is it can facilitate in the organization of chromatin, RNA polymerase 2. For example, in this example, if you look at it here, fire RNA allows the formation of a secondary allows the enhancer element to bring together near the different chromosome. It assembles the structure in such a way the RNA polymerase comes together at a transcriptional unit. Then this kind of loop formation, if you have CTCF, MIG promoter, all these things are mediated by long non-coding RNA, and transcribed, for example, the MIG promoter region is involved in the secondary structure formation. Then another way is at a nucleosome organization, as we talked about. Again, what is happening is that, as we talked, cis RNA compacts it and recruits through other long non-coding RNA, the chromatin remodeling complex twice NAV complexes can be recruited and change the organization of the chromatin itself. The long non-coding RNA can also bring about change in the chromatin structure by forming R loops. So R loops are very critical in the recruitment of RNA polymerase 2, thereby changing the transcriptional level. And I think in earlier class, when we were talking about regulation of the regulation by RNA gene expression regulation, the long non-coding RNAs can sequester, for example, in ALU or SINE sequences, the short uh, uh, elements. So this can erase the RNA polymerase at the initiation stage and doesn't allow it to transcribe these repetitive elements. And many a times, for example, in this case, at the elongation step, it can defect as DXD2 along with 7-ASK. The RNA polymerase is blocked at the elongation step. And long non-coding RNAs are very much involved. Yeah, do you have any questions? Okay, so the again, the long non-coding RNAs are also involved in uh, organizing granules in the RNA, and many times in organizing the RNA retention as well, and in organizing intronic region or splicing, all known as the splicing, then microRNA modulation, So there are different the long non-coding RNAs can act in different level of gene expression. Okay, so I think with this I'll stop for today. But next class we will go with a little longer class. Okay, so
अभी तो सरू का वेडिंग रिसेप्शन देना बाकी है। इन्हें कब डिस्चार्ज हो सकते हैं? और इंडिया से पूरा परिवार को बुलाना है। और इसका बहन वो यूएस का है, उसको भी तो बुलाना है। टाइम लगता है ना? हाँ?
so I am planning to send a video as well as most of the things are in the most of them are there in the reference that I have sent earlier, in the papers that I shared earlier. Okay, so if you read those shared material, that should be good enough for you people to cope up with this one. Okay, so yes, definitely that definitely uh, the slides I will be sharing. Okay, slides.
इसका क्रिया कर दो और ये सब
Okay. Uh,
कौन मदद अपने बच्चे के लिए ऐसा कहता है?
we also share a small ungraded assignment. Yeah, we can think about it. For the assignment, ungraded assignment, we can think about
ये तुमने
definitely. So the some of the sections we need to discuss is the uh, application of molecular biology, something like protein expression, then cloning. All these things uh, we need to do. I think we can do it in an assignment mode. We can discuss it, and we can do in an assignment mode as well. How to clone. Maybe we can discuss one day. Just sit down and discuss. Okay. So that's it. I'll stop here for today. If you have any questions, let me know. So regarding there is a question from Akshay J. Can certain non-coding RNAs be used to trace evolutionary relationship? I don't have very specific example, but I guess so because the transcriptional units, many of them, for example, the non-coding RNAs, which are very similar to humans or very specific to humans, have certain similarity with the chimpanzees, or which is the closest to humans is have been seen, not with any other primates. So I think definitely the non-coding RNAs can be used as evolutionary tracking machinery. I guess so, because only those close ones will be sharing certain more long non-coding RNA conservation rather than the uh, I think so. We can track the evolutionary relationship. But I think computationally it has to be quite complex, I guess so, because only 70–80% how much conservation is there, I'm not very sure. So definitely it can be used. Okay. So Yas Palley is talking about ungraded assignment for every lecture. So that we can think about maybe write something about some of the regulatory elements. Maybe I can send out in a group, maybe because I can't be looking assignment every week, 90 of you. So maybe we can group together. You talk to each other and then uh, write the proposals. You can divide into segments or something. That definitely we can do. Maybe A to. Okay. So I think Himanshu has good suggestions. I think maybe I will ask share some questions to answer, ungraded ones that I can do. I will do in the Google Forms or something that I can share with all of you, and then you people can respond to it and then see how well the response happened. I think this one, Himanshu's one is very good, I think. Yeah, this will be good. I think Himanshu's suggestion is very good. Uh, yeah, this is good actually. So, yeah, we can do this way. Definitely, maybe around for each class, maybe at least around 10 questions or something we can think about and then go ahead as an assignment. Okay, yeah. Any more questions? Okay, so for minors, we have more than enough work till now. Please consider also. So, Rezit, what do you mean by you don't want any assignment, or what is that idea? Yeah, can you elaborate more? Okay, I think Himanshu's one is if it is ungraded, if you don't want to attempt, maybe quite okay, reasonable. If you want to try, also okay. Okay, maybe we'll just form certain questions and then discuss in the next class. That's what we'll do. I think I'll send out Google Forms. Some of you can respond, or you can choose not to respond. Okay, so maybe that is a good idea. I think we'll go easy. Not it's not going to be it's not going to be graded. We'll just have if some of you can respond. Otherwise, we'll just discuss the questions. Yeah. Is that okay? Okay. So any more questions? Okay, then I'm stopping. Okay. See you in the next class. Okay.
Okay, thank you, sir.
Yeah. Mm.